For those of you who care about or participate in the LARC program, it's probably worth going to this event on the 22nd. So let me pick up again where we left off on Wednesday and review a little bit. On Wednesday we talked about the definition of species, and I said that species were evolutionarily independent units. That is, the evolution of two different populations where mutations arise independently in each and adaptation occurs independently and their future trajectory is independent. Those are different species. Now, we talked about different ways that species can be defined. We talked about the biological species concept. Things are defined as being in different biological species if they do not interbreed. And we talked about some of the difficulties of applying that definition. We talked about the morphological species concept. This is what you apply mostly to fossils, for example, because that's all you've got is the morphology of the fossil to look at. And that's simply based upon there being sufficient morphological differences to feel that they are evolutionarily independent populations. And we talked about the phylogenetic species concept. This is where species are actually defined as the terminal tips on a phylogenetic tree. Then we went on to consider the concept of subspecies, and I said that subspecies don't have the kind of objective definition that biological species do. And we used as an example of subspecies Neanderthals and humans, and we talked about the fact that these were two different groups which had evolved independently for a long period of time. Modern humans did not come up into Europe until 60 or 70,000 years ago, so they really weren't contacting Neanderthal populations until after Neanderthals had been in Europe for close to half a million years, for 500,000 years or more probably. And yet there was still the possibility of interbreeding, and we know that because we can look now at Neanderthal DNA and sequence it from fossilized bones, and we can identify Neanderthal genes in the human genome, and we know that somewhere between 1 and 4% of the genes in the human genome have a Neanderthal origin, indicating the fact that there was some genetic exchange, some hybridization between Neanderthals and humans. So subspecies are populations which are on the way to being species, but they haven't reached the point where they are genetically isolated. Now we're going to talk about the common processes that lead to new species. And most of the time, speciation occurs as a geographic process. Populations get isolated geographically, and so there is no gene exchange simply because they're isolated. And that's called the allopatric speciation model. So that's divergence under allopatry. Allopatry just has Greek roots meaning different regions, different countries. And there are a number of different ways that isolation, geographic isolation, might arise. There are two that are discussed in your textbook. One is simply dispersal. We saw lots of examples already of dispersal, particularly with regard to island situations. We talked about the Galapagos Islands and the fact that species were different on different islands because they'd been isolated. Well, that's an example of dispersal, populations coming to an island, colonizing it, but being geographically isolated from the parental population. If this goes on long enough in time, then they eventually are reproductively isolated. The other is just a different variant on the same idea. This is called the Vicariance process, and that's simply where there's some other kind of extrinsic geographic barrier between populations, like a river or a mountain range, or we'll see an example where continental drift plays a role in this vicariance process. So it's some other kind of extrinsic barrier between populations that provides a physical barrier to genetic exchange. So this figure from your textbook just illustrates these two ideas. The dispersal hypothesis, you have individuals that leave the parent population, colonize some other region, and are geographically isolated. Maybe they colonize an island and therefore are physically, geographically isolated. They evolve independently for a long enough period of time where they accumulate genetic and eventually morphological differences. The vicariance model, there's some sort of separation. Maybe it's a river, a large river that separates two populations, and they remain separated, and they can't cross the river, so they can't mate. And over a long period of time, mutation, genetic drift, and natural selection for different adaptations in the two regions lead to the accumulation of sufficient differences that they're recognized as different species. So here's an example of a vicariance process. This one's not in your book. It was actually in the previous edition, and I kind of like it. It's an example taken from continental drift. About 150 years ago, there was one massive continent on the Earth. It was called Gondwanaland. And during that period, a group of flightless birds called Ratite birds began to evolve, and presumably there was a single species, but then over time, the continental plates began to break up and drift on the surface of the Earth, and those provided physical separations that led to the differentiation of Ratite populations, which ended up on different continental plates until at the present time we have a series of Ratite birds. We have the ostriches, which originated in Africa, emus in Australia, kiwis in New Zealand, and so forth. These are all evolutionary lineages which have been isolated over a very long period of time by the physical processes of continental drift. So that's a vicariance kind of explanation for the isolation of the populations. But the key idea is that if there's enough time that goes by, drift, mutation, and natural selection for specific adaptations in separated populations will lead to genetic divergence. If there's no opportunity for genetic exchange, that divergence accumulates through time, and ultimately the populations become sufficiently different that they're recognized as new species. Now, the question arises, is it possible to have speciation occur when the populations actually overlap geographically, when they share some of the same physical space? And the answer is not very often. It's a rare phenomenon, but it can happen, and it's called sympatric speciation, described here. So can populations diverge to the point of becoming new species when they are in physical contact, that is, sharing the same space? Sympatric speciation. Well, there's a really nice example in your textbook of the process that might lead to sympatric speciation, and this is actually the apple and hawthorn fly example. It turns out that apples were introduced into North America by European settlers about 300 years ago. They didn't exist in North America prior to that. The apples are a domesticate of Central Asia, and so apples didn't exist in North America before Europeans brought them there about 300 years ago. But there's an apple fly which specializes on actually the flies congregate on apples, they mate, the female deposits her eggs in the apple, the eggs mature, the larvae eat the apple. As the apple ripens, it falls to the ground, then the larvae actually crawl out of the apple into the ground, they pupate, and a new generation of flies emerges. So they're specialized on apple fruit. But when the ability to look at DNA developed, some investigators asked, where did the apple fly come from? Did it come with the apples, or did it emerge as a new population de novo in North America? Well, it turns out that it's genetically almost identical to the hawthorn fly. Hawthorns are native to North America. They also produce a fruit, and the hawthorn fly has a similar life cycle. The flies congregate on the fruit, they mate, the eggs are deposited on the fruit, the larvae emerge, they subsist off the fruit fall to the ground, and so forth. So it's a very similar life cycle. But what's happened is that in the 300 years since the introduction of apples into North America, there's been a divergence of two populations that originated from the hawthorn fly. And one group has specialized on apples, but its origin was in the hawthorn flies. However, if you now test apple flies and hawthorn flies, you can show that they distinguish between apples and hawthorns by scent, by odor. And apple flies only go to apples, and that's heritable. Hawthorn flies only go to hawthorns, and if you intermate them, they don't know where to go, okay? So in this short 300-year period, there's been a divergence between two populations, the apple fly population, which originally derived from hawthorn flies, and they're becoming more distinct genetically. So this is a possibility for sympatric speciation, because the two trees live in the same environments close by. And of course, the fact that the mating occurs on the fruit means that there's very little opportunity for intermating between apple flies and hawthorn flies, because they're specializing on two different discrete resources in the environment. So this is one example of the possibility for sympatric speciation, but generally speaking, the geographic model of speciation is what probably happens most of the time. There is one exception, which is quite important in plants, and that's called polyploidy. Polyploidy is a situation where, you know, that when you produce a gamete, it's haploid. It's one copy of each chromosome in your genome. Okay? You've got 23 pairs of chromosomes. Those separate at meiosis, so that one copy, one of the two parental copies, is segregated into a gamete to provide a complement of 23 in the gamete. That's a haploid gamete. Occasionally, something goes wrong with meiosis. And instead of the proper distribution of the two parental chromosomes to the different poles of the cell, they all end up in one pole. And so you produce, I'm using you in a metaphorical sense, you produce a gamete which is diploid rather than haploid. Okay? Just an error, it's a big mistake that happens in the cellular machinery. If two diploid gametes unite, they will form a new tetraploid individual. It's not diploid now, it's tetraploid. It has 4 copies of each of the parental chromosomes rather than 2. Okay? That's a tetraploid. So that's one example of polyploidy. And this figure sort of provides a graphic illustration of it. Polyploidy is really important in plants. We don't see it very often in animals, but it has been important in the evolution of some lineages of fish, but it's really important in plants. The majority of grasses, for example, have a polyploid origin. So polyploidy has played a big role in plant evolution. And this is just a diagram of the process I described. You have the diploid parent. Typically, it produces a haploid gamete, but sometimes there's a tetraploid gamete. If those unite to form a new individual, you have a tetraploid individual. And so now you ask, what happens if this new tetraploid individual mates with a diploid individual in the population? Well, one gamete is going to be diploid and the other is going to be haploid, right? And that's what's shown here. So you have a haploid gamete uniting with a diploid gamete, and that produces an individual who has 3 copies of each chromosome. Okay? Well, 3 copies of each chromosome presents a problem in meiosis. You can't have the regular segregation of the chromosomes in meiosis. And so what occurs is that the hybrid offspring are almost always sterile because the gametes have uneven chromosome numbers. Okay? It's very simple. Now, do you know of any examples of triploids in the world that we live in? You eat them frequently. The navel orange is a triploid. It's a triploid, and that's why there are no seeds. It doesn't, because during the process of seed development, they degenerate because they don't produce a zygote, which is viable. So the navel orange is one example of a triploid. Another example are the Thompson seedless grapes. Well, how do they propagate these? If they're going to not produce seed, how do they get propagated? Well, they get propagated asexually. They get propagated because you can take a graft of the plant and repeat it many times and grow them asexually. But the navel orange and the Thompson seedless grape are 2 good examples of triploids. But from our point of view, from the evolutionary point of view, what's happened here is that there's an instant reproductive isolating barrier between the diploid population and the tetraploid population. They can't mate and produce fertile offspring. So there's an instant post-zygotic, remember we said pre-zygotic or post-zygotic reproductive isolating barriers? This is post-zygotic. An instant post-zygotic reproductive isolating barrier between the tetraploid population and the diploid population because they can't produce viable offspring. And it happens a lot in plants. Now, why is it likely to happen more in plants? Well, there are 2 answers to that question. One is that plants have what is called an indeterminate growth structure. The meristematic tissue, which is the growing tissue of plants, grows out at the tips of stems, and eventually there's a genetic signal that it should develop into a leaf or into a reproductive structure, and that takes place. But this is after a long series of mitotic divisions in the meristematic tissue, and then more mitotic divisions as it differentiates into, let's say, a reproductive structure. Well, if somewhere in that process during mitosis there was an error that led to a doubling of the chromosome number because there wasn't an appropriate segregation of chromosomes to the 2 poles, then you have potentially a whole clone of tissue which are tetraploid. So you end up with a plant that has some of its tissue that's tetraploid, some of it is diploid. If some of the tetraploid tissue differentiates into flowers, it'll produce gametes, pollen and eggs, which are diploid, not haploid, okay? And that happens not infrequently in plants. And the other thing that happens in plants that facilitates the production of polyploids is that many plants self-fertilize. Remember, we talked about self-fertilization. We saw the consequences of self-fertilization. But if you have a plant that has a sector that's producing some tetraploid gametes, and they can self-fertilize, then you have the potential of producing new tetraploid individuals from what was originally a diploid parent. That happens frequently enough in plants that polyploidy is an important mechanism of speciation in plants. Okay, now, let's suppose that we've got two isolated populations which are close to being different species, and we bring them back into contact. What's going to happen? Well, if prezygotic isolation exists, there's no possibility for the union of gametes, then they're going to continue to diverge, right? Because there's already prezygotic isolation. They're not going to produce any offspring, so they're going to continue to diverge. What if that's not the case? What if they still have like the Neanderthals and humans? What if they can still intermate and produce offspring? What might happen then? Well, there are a series of different things that can happen. Most likely, the hybrids, if they've been separated for a long enough time, in most cases, the hybrids are going to be less adapted than either of the two parental types. And that's because the parental types are adapted to environments which differ in different specific ways, and the hybrid is not going to be adapted to either of those environments. Just as we discussed in the case of the apple fly and the hawthorn fly. If the hybrid has a disadvantage, then there can be a selection called reinforcement selection operating against hybridization, because the reproductive output of both of the two parental populations is diminished if hybridization can occur. So there's just going to be natural selection opposing the possibility of intermating. There are two other possibilities. One is that you could produce an entirely new and novel species called a hybrid species. And that does happen very, very occasionally. And the third possibility is that you produce something which is a hybrid zone between the two populations. It can persist for some time, and the hybrid individuals are recognizably different, but it may not be stable. And here's an example from the textbook. This is along the northern Pacific coast from southern Alaska down to the state of Washington. And the red shows the geographic distribution of a species of warbler called Townsend's hermit. And the middle one is a hybrid of the Townsend's hermit with the Townsend's warbler with the hermit warbler. The middle one is their hybrid. The Townsend's warbler is a much more aggressive bird. It defends territories, it is aggressive in seeking mates and so forth, than is the hermit warbler. And there is speculation that the geographic range of the Townsend's warbler is gradually moving southwards into the hermit warbler territory. Where the two have come into contact, there is another stable population of hybrids, which is the middle type. Now, the ultimate fate of that hybrid population is hard to predict. Probably most likely, certainly if the Townsend's warbler eventually replaces the hermit's warbler, there will no longer be any hybrids, and the hybrid population will be a transient phenomenon. But hybrid zones, as in this case, are a third possible outcome of hybridization. Okay, so when when isolated populations come into contact, there's a simple set of possibilities. Prezygotic isolation exists, and they'll continue to evolve independently. There's unlikely to be any gene flow between the populations, and they diverge. If, on the other hand, gene flow occurs, if it's possible, then there's also two possibilities. One is that gene flow will eventually swamp the two different populations, and you'll end up with one population, which is the average of the two original ones. The other is the development of hybrid zones or reinforcement. And reinforcement, as I have already told you, is simply natural selection operating against intermating, because intermating reduces the reproductive potential when the hybrids are inferior to the two parental types. So that is, that concludes then this part on the way in which species arise, the kind of evolutionary dynamics of species. And we're going to go to the quiz. And so you'll need to put away all your books. We're going to give you 1 minute per question this time. But you need to put away all your books and computers and everything. And I'll turn it over to Kate, who will run the quiz.
Yeah.
So, soon as everybody's got their materials put away and you're ready to go, we'll start.
Yeah.
Do you have your stuff all loaded? So
I'm sorry? Yeah, it's all,
okay,
all ready
okay.
to go. So whenever everybody is ready again.
Is everybody happy? It's Friday. It'll be a 3-day weekend. This is an easy quiz. Are we ready? Okay, so here's the first question. And you've got 1 minute. Okay. Ready for question number 2? Okay. Okay, there we are, number 2. Okay. And finally, number 3. Okay, is there anybody whose clicker didn't work and they had to write? Can you, yeah, let us collect those? And you remember to put your name and your student number on those, otherwise we won't know who you were. Okay, if we got them all, well, one more. Okay. This, you know, it's too many people with non-functional clickers. We won't be able to do this for the midterm, so you'll have a big problem on the midterm if your clicker isn't working. Yeah. Yeah. I'm sorry? I'm sorry?
For the midterm, we're not using our iClicker, are we?
I'm not sure whether we are or not, actually. Scantron, okay, be okay. Okay. Okay, so let's quickly look at the answers. So number A, genetic correlation occurs when selection favoring alleles for one trait cause a correlated but suboptimal change in allele for another trait. True, that's right out of the book, and it's also the definition in class. Fitness trade-off is a compromise between traits in terms of how those traits are adapted to the environment. That's also true. It's again directly from the stuff in class, and it's in the book. C, all traits evolve from previously existing traits. Adaptations are constrained by history. True. Again, directly from both the class and the book. Environmental changes can result in changes in the phenotypes that are favored by selection. Yeah, of course. Selections, the environment is the ultimate selective agent. So in the case of selection for the del32 mutation in CCR5 gene is an example of disruptive selection. No, it's an example of directional selection. It's not disruptive. In fact, remember we said that one homozygote has a very large disadvantage because people die fairly fast of HIV/AIDS, and the heterozygote is intermediate. That's directional selection. Okay, choose the incorrect answer regarding the Hardy-Weinberg law. It shows that allele frequencies do not change under random mating. True, that's something we emphasized. It establishes the conservation of genetic variance principle. True, can be used as a null hypothesis to test for selection. That's true, we've used that. It is true for inbreeding as well as random mating. False, because we showed you that the genotypic frequencies change under inbreeding, and we went through that example of self-fertilization. And then here's a little calculation, which was designed to be one you can do in your head. 0.9 squared is 0.81 and so forth, okay? And then number 3, I don't seem to have control. Okay, this is number 3. Which of the following statements about genetic drift are false? The effects of genetic drift are more pronounced in large populations. Well, that's false. We went to great effort to demonstrate for you that genetic drift is most prominent in small populations, and the larger the population gets, the less the drift effect is. Genetic drift is a random force, absolutely true. Genetic drift is a non-adaptive evolutionary force, that's true. Genetic drift ultimately leads to the loss of genetic variation, that is true. Mutations that do not affect fitness will ultimately be fixed or lost. That is true. So that's the answers to number 3. I don't know if we can show the histograms of.
What you did? Okay.
Okay.
So this is for question 1, distribution, and the correct answer was E. Yeah. Question 2, correct answer was D. And question 3, the correct answer was
A, yeah. Okay, so we need to look at question 1. I'm kind of curious that so many of you got it wrong, but let me now move on to the topic of phylogenies and the fossil record. Okay, we want to make inferences about the past. A lot of evolutionary biology is about making inferences about the past, and we really have a very powerful array of tools for doing that. One set of tools is by building phylogenetic trees, by inferring past genealogical relationships among living organisms. Who's related most closely to whom? That's a phylogenetic tree. The other is the fossil record, and we've already talked a little bit about the fossil record. We'll talk more about that in this lecture. Here's a phylogenetic tree. This is one I showed you in the first lecture, and in fact it's the tree of life with the 3 domains, okay? Now, this is a very abbreviated tree of life. It would be huge if we tried to get right down to individual species, most of which we don't know where they place exactly. But there's some features to point out. One is that we refer to the lines that go out to the named groups at the end. Named groups at the ends are generally called taxa, that's plural, taxon is singular. The lines that go out to them are called a branch. We just use an analogy to a tree. These are the branches. The places where 2 branches connect is a node. The end of the branches is called a tip. And a tree can be rooted in time if it's oriented in time. So we have 2 ways we can depict a tree. One is it's oriented in time. This one, the node at the bottom, represents the common ancestor of all life on Earth, and this is oriented in time. But sometimes we don't know enough about time, about who's oldest on a tree to orient it in time. There is an approach to doing that I'll just hint at. So branches, 2 adjacent branches are called sister taxa. Taxon on the end if it's singular. The tips are the end points. They represent living groups or groups that end in extinction. And the names at the tips may represent species or larger groups. They represented much larger groups than species in the picture I just showed you. It had plants and animals as endpoints. Their nodes, I've already told you. A polytomy is only where there are 3 branches that go off from the same node. Okay? A rooted tree, that's one that's oriented in time. I've already mentioned that. And we actually try and determine how to orient them in time by using what's called an outgroup. That's the oldest. That's some taxon which is older than everything else in the tree, and that tells us where the deepest node is in time. But we can't do that for the tree of life because there isn't anything older than the ancestor of all life on Earth that we know about. So we have to use different approaches to try and orient the tree of life in time. There are 2 ways in which, very broadly speaking, there are 2 different approaches that are used to building these trees. I'm just going to tell you a little bit about them, just so that you have a flavor for it. One is called Frentic methods. These just use some statistic which measures the degree of similarity or difference between populations as a basis for clustering them and building up a tree. And we'll just quickly look at a simple example. And then the other approach is called cladistic. Now, for the Frentic approach, let's just think about an example. We've got 4 species, A, B, C, and D, and we have measured them for some quality. So we can make a 4x4 matrix, and in each cell is the value for either the similarity between 2 species for that quality. So here's an example. This is the matrix. Species A is the similarity measure between A and B is 3, between A and C is 5, between A and D is 8. Okay? So clearly A is more similar to B than it is to C or D by this measure, whatever it is. But we start with the pair that are closest, that are most similar. What is that? Well, that's those with the 1 in them. That happens to be C with B. C with B C with B are most similar. So we cluster those. They're differ by only a single unit, so the leg up from C to the node is a half, and then a half down to B, and that gives us our first node on the tree, and then we go to the next most similar one and calculate its similarity to the average of C and B. Well, the next one is A, I guess. So we group A in, and we just do it systematically that way, so that when we're finished, we have built a tree that depicts the relationships based upon their degree of similarity for this measure, or degree of difference. Either way, they're complementary. That's the Frantic approach to building trees. Now, the other approach is called the cladistic approach, and this is actually based on inferring trees based on shared common traits, shared ancestry. So these are traits which are believed to have been derived from a shared ancestor that are shared by 2 or more taxa. Group those taxa together. And those traits have a name. They're called synaptomorphies in this business. Unfortunately, there's a lot of terminology, but anyway, they have a name and they're called synaptomorphies. And this is an example of, using DNA sequence data, of grouping together taxa based on traits that are shared by descent. So we go back to the ancestral, the common sequence at point A, that's AA, GCT, ACT. This separated into 2 different lineages, and in one lineage, lineage B, there was a mutation to C at the 3rd position. And that mutation is shared by species 1 and 2, so that groups them together in history. On the other lineage, there was a mutation at the 5th position, a C to a G. And that is also shared, that G mutation, by the species 3 and 4. Okay, so that's the process. Now, it's not done for just one trait or one mutation like this. It's done for lots of mutations, and you take the results which have the highest number of shared derived characters, or synaptomorphies, as representing those which derive from a common ancestor. So that's how these trees are built up, okay? That's how the inference is actually carried out. I'm not going to spend much more time on this because I don't think it would require much more detail than is merited for the level of the class. Okay, but there are problems that can arise with these approaches. One problem is convergent evolution. What happens if over time you have populations which were very different, but they become adapted to similar environments again, and in ways that lead to similar morphology? Then that's going to fool us. That's going to fool us, because we're going to score them as being more similar than they really are. And we'll see an example in the next slide. So there are 2 other terms or concepts that are introduced. One is homoplasy. That refers to traits that are similar for reasons other than common ancestry. And the other is homology. These are traits that are similar due to shared ancestry, okay? So homoplasy will fool us. Homology is what we want to understand. That's shared ancestry, so that we can build a tree that really reflects the true evolutionary history. And here's a nice example from your book, which is an example of convergent evolution, and it's the dolphin versus the ichthyosaur. And they really look very, very similar. They're both swimming animals, sharp teeth, streamlined bodies, long jaws. They really look very similar, but they're quite different forms of life. One's a reptile and the other is a mammal. And you can see that the dolphin actually evolved independently from a lineage most closely related to marsupials, whereas the ichthyosaur is most closely related to lizards. So they evolved from very different parts of the evolutionary tree, but because they adapted to a similar marine environment, they converged in morphology. That selection actually resulted in this convergent evolution. And it's only when you look at underlying traits like DNA, or perhaps in this case some features of skeletal anatomy and so forth, that you realize that these are really very different. Here's an example of homology, also from your book. This is from a family of genes that are really important. They actually are fundamental in determining body plan in insects, humans, even in worms. And they're called the Hox genes. And they built up over evolutionary time. There have been a number of duplications of Hox genes. The ones with the same color have an origin through duplication. But the two genes, the top represents an arthropod, an insect, actually the Drosophila Hox gene cluster, and the bottom represents the human Hox gene complex. And these are clearly homologous. They derive from a common ancestor long in the past when the first evolution of segmentation and body plan began to take place. So this is an example of a homologous trait. And in fact, if you sequence the Hox genes, you further establish that they're homologous. Okay, I guess this is a good place to stop, so we're going to come back and talk about whales on Wednesday. Have a great 3-day weekend.